Today we're going to go through mitosis. So just as a recap from lecture, what is the role of mitosis? Why do it at all?
To duplicate cells?
Yeah, to duplicate cells. So where does that become helpful? Like what kind of scenarios?
To maintain their survival of reproducing.
For growth?
For growth, yeah. So growth is one of them. And then what's the other one? Reproduction. So growth and repair. Yeah, so Ryan got that one right. So we're going to use it for helping us be able to grow. And then we're also going to use mitosis to help us repair. So today we're going to be focusing on mitosis. There's also a separate process that's also in this lab called meiosis. Meiosis is more for actual like allowing sexual reproduction. So we'll look at that next week in lecture. And it's going to follow the same general phases of mitosis. For the sake of today, we're just going to be focusing mainly on mitosis, and there's a couple of videos on meiosis in the lab itself. So we're going to run through this. So again, mitosis is going to start a phase called interphase, and that's going to be where our cells are going to be growing. They're going to be replicating their DNA, and then they're going to be accumulating all of the energy that's going to be necessary for them to be able to begin to divide. Okay. Once they go out of interphase, they're going to enter prophase. And so in prophase, they're going to duplicate, or they're going to have their centrosomes already duplicated, and they're going to start producing their microtubules, their mitotic spindles, and our chromosomes are just going to be kind of all over the place here. Okay. So in prophase, so in prophase, then we have metaphase, which is going to be where our chromosomes are going to line up. We're going to have anaphase, which is where our chromosomes are going to split, and then we'll have telophase at the end of this. So just to kind of go over everything here. So mitosis, or the mitotic phase of mitosis is going to be, or actually, yeah, let me just backtrack. So mitotic phase is important to help the cells and their nuclei be able to split apart and then divide it into two genetically identical daughter cells. Okay, so remember, that's the key. We want to produce identical daughter cells here. So there's two phases of the mitotic phase. The first one is mitosis, and that's going to be where our duplicated chromosomes, which have been duplicated from interphase, they're going to get aligned, they're going to get separated, and they're going to get moved to opposite ends of the cell. Okay. And so it's going to happen typically in about 4 stages. Some biologists consider a 5th stage, which is prometaphase. It's really just an in-between, like an intermediate stage between prophase and metaphase. For the sake of our lab, we're going to just acknowledge that it's there, but we're not really going to do too much with it. But essentially, again, the goal of mitosis is to just move the chromosomes to opposite ends of the cell. Okay. Then we have the second phase of the mitotic phase, which is cytokinesis. And that's where we're going to actually separate that cell into 2 individual daughter cells. Okay. So we're going to kind of go through the different phases, and we talked about it in lecture, so I'm not going to beat you guys over the head with it too much. But if you have any questions, again, just feel free to stop me, and then we can kind of explain this a little bit in more detail. And so the first phase is prophase. And so in prophase, what we need to do is we need to start accessing our nucleus, and we need to get it to be easily accessible. So the first thing that we're going to do is we're going to eliminate the nuclear envelope and cause that to break down. We're going to see a Golgi apparatus and then an endoplasmic reticulum. And so because those guys also surround the nucleus, we're going to break those down as well and then move them away from the nucleus. And then there's going to be a nucleolus within the cell that's going to end up disappearing as well. The next thing that's going to happen is we're going to have some microtubules, which are going to end up making our mitotic spindles. They're going to start being produced by the centrosomes. And when they get produced, they're going to latch onto the chromosomes. And they're going to bind to the centrosomes, specifically here at the middle part of your chromosomes. Okay. Once they start getting produced, they're going to begin moving the centrosomes to opposite poles of the cell. And then we'll also start to see the sister chromatids of each individual chromosome getting becoming more visible. Okay. So if we remember from looking from lecture, this whole structure here would be considered one chromosome. Okay. And then each of these legs, so kind of circle, let me do a different color. Each of these legs here would be considered one sister chromatid. So at this point, we have two sister chromatids making up our chromosomes. Okay, cool. Following me so far? All good? Okay, so now we are going to move into the next phase, which is, this is pro-metaphase. And so again, some biologists will consider pro-metaphase to be a phase, other ones won't. I'll just kind of go through it, but for the sake of our class, we're not gonna really worry about pro-metaphase. Essentially, pro-metaphase is, like I said, an intermediate phase between prophase and metaphase. And so basically everything is going to continue. Prophase is going to continue like normal. The nuclear envelope is going to continue to disappear and break down. And then the mitotic spindles are going to begin to connect to the to the chromosomes at the centrosome. Okay? And specifically, they're going to bind to the kinetochore. Okay? So as we see here, each sister chromatid is going to have a kinetochore on it. Okay? So each kinetochore for each chromosome needs to be attached to a mitotic spindle. Okay? And so that's for the that's going to be one thing that's going to be important to keep straight here. So the mitotic spindles are going to begin to bind at the kinetochores on the chromosome centromere. Okay, then we move to the next phase, which is metaphase. Okay, and so in metaphase, the mitotic spindles, which are now bound to all of the chromosomes, are now going to cause the chromosomes to line up at the metaphase plate. Okay, and so this metaphase plate here is going to be oriented at the equator of your cell, okay, basically at the midpoint of the cell, and they're going to line up in a single file line. Okay, the sister chromatids are still going to be attached, and then at this point, the chromosomes are going to be fully condensed here. Okay, so again, what's going to be important here is making sure that the mitotic spindles are attached to all of the kinetochores. Okay? And so that's where the M checkpoint comes in that we talked about in lecture today. So at the M checkpoint, we're going to be checking to make sure that all of the chromosomes have been attached to a mitotic spindle, okay, at the kinetochores, and they're going to be lined up here at along the metaphase plate. Then we have our next phase, which is anaphase. And so in anaphase, what we're going to have is our sister chromatids getting separated from our from one another. Okay? So here in metaphase, we have our chromosomes all lined up along the metaphase plate. Okay? In anaphase, the mitotic spindles are going to pull the sister chromatids apart, okay, and pull them to opposite ends of the cell. Okay? Again, closer to the centrosome that it's attached to. And then also in this phase, the cell is going to elongate. So again, over here, we can kind of see our cell beginning to stretch while our chromosomes are being pulled apart. Okay. And then the last phase is telophase. Okay, so in telophase, we have our chromosomes that have been pulled to opposite ends of the cell. And so now what we want to do is we want to start kind of restoring the original conditions of the cell. And so we're basically going to be undoing everything that we did in prophase. Okay? So the first thing that's going to be happening is our chromosomes are going to decondense. So initially in prophase, the chromosome, and in interphase to a lesser degree, but our chromosomes began to condense. Okay? In telophase, what we're going to do is we're going to start decondensing our chromosomes. Okay? And so we'll see the role of that in a couple of weeks. Okay? So that's number one. So chromosomes are going to decondense. Number two is that the mitotic spindles that we produce from the centrosomes are going to start to break down. And then from there, those little components of the mitotic spindles can be used to form the daughter cell cytoskeleton, okay, and give that cell its own shape. Okay. And then the last thing that's going to happen is our nuclear envelopes are going to reform around all of our chromosomes here. Okay. And so what we're going to do is what we're going to end up with is two individual nuclei inside of a single cell in telophase. Okay. Questions so far? Okay. So that is mitosis. All right. That's mitosis. Okay. So again, the point that comes after telophase is this phase called cytokinesis. Okay. So remember, at this point, we have one single cell with two nuclei in it. Okay. We don't want that to happen at the end of this process. We want to end up having two individual cells. Okay. So we're going to have this process called cytokinesis, which is going to be where we're actually going to physically separate those or that cell and split it in half to end up forming our two daughter cells. Okay. And then again, depending on whether we're looking at a cell that has a cell wall or not, okay, this process is going to look a little bit different. Okay. So if we're looking at animal cells, which do not have a cell wall, cytokinesis is going to start in anaphase. Okay. And so what's going to happen is we're going to have the cytoskeletal filament called the actin filaments. And so what actin will do is it's going to form a ring around the metaphase plate, okay? Kind of like just a little ring, right? Just like this. Okay. Once we form that ring, once actin forms that ring around the metaphase plate, actin is going to start to contract. And when it contracts, it's going to cause a little fissure right here in our metaphase plate. Okay. So we're going to see a little groove start to develop here, a little fissure here. Okay. So when this happens, this is called the cleavage furrow. Okay. And so the cleavage furrow is going to continue to constrict and constrict and constrict and constrict and constrict until you end up splitting your cells in two. Okay. And so that's exactly what we see here. Okay. We see our cleavage furrow constricting, 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 constricting until you eventually split that cell in two, and each cell now has its own nucleus. Okay. So initially we started off with one single cell with one nucleus here and another nucleus here. Okay. At the end of cytokinesis, we eventually split this in half, and then you end up with two daughter cells at the end of this whole process. Okay. And so this is what's going to happen in animal cells. So humans and then any other animal that you can think of, these are all going to perform cytokinesis in this particular manner. Okay. Anything that does not have a cell wall. Okay. Now, if we're looking at a plant that does have a cell wall, this process is going to look different. And the reason for that is because the cleavage furrow cannot pinch the cell wall. The cell wall is very, very strong and very rigid. And so actin has really no way of splitting that cell in half. Okay. So instead, what we need to do is we need to create a new wall in between the two nuclei. Okay. And so the way this is going to work is when we go back to interface, and then if we rewind back to interface, the Golgi apparatus is going to start to accumulate a bunch of enzymes and structural proteins and glucose. Okay. When they start to accumulate all of that stuff, they're going to store it inside of these vesicles before the Golgi apparatus breaks apart in prophase, okay, like we saw earlier. Okay. So when it starts to break apart, those vesicles containing all those enzymes, structural proteins, and glucose are all going to be scattered around the periphery of the cell. Okay. So then now if we fast forward into telophase, okay, once we get to telophase, the Golgi vesicles are going to end up coming back into the metaphase plate. And they're going to line up right along those, or they're going to come along these microtubules and just line up here along the metaphase plate. Okay. Once they fuse here, those vesicles are, or once they arrive there, they're going to start to fuse. And when they fuse, they're going to be joining a lot of those structural proteins together, a lot of that glucose and a lot of those enzymes to help form a structure called the cell plate, okay, in between the two nuclei. Okay. And so the cell plate comes from the vesicles all fusing with one another. Okay. Eventually, it's going to continue to enlarge and enlarge and enlarge until it eventually fuses with the cell wall itself. Okay. So once it fuses with the cell wall, okay, eventually that will eventually form its own cell wall that actually physically separates this cell into two individual plant cells. Okay. And a lot of the enzymes involved are going to help take glucose and then link a lot of those glucoses together to help form cellulose, which is what makes up plant cell walls. Okay. All right, so that is cytokinesis. Okay. Okay, so that's the whole process of mitosis. Again, we're starting with interphase. Okay. With interphase, or after interphase, we're eventually going to go into prophase, where we have our chromosomes all over the place. We've destroyed our nuclear envelope and gotten rid of a lot of those Golgi apparatus and the ER. From there, we're going to enter metaphase, where we have our chromosomes line up along the metaphase plate, okay, by the microtubules. Okay. In anaphase, the mitotic spindles will then pull those sister chromatids apart. Okay. And remember, at that point, each sister chromatid is now referred to as its own individual chromosome. Okay. So now they've been pulled apart. And then in telophase, we're going to reform that nucleus around around each of the chromosomes. And then the last thing that's going to happen is we're going to have cytokinesis, which is going to split this cell in half. Okay? And so that's going to ultimately form 2 daughter cells at the end of this process here. Cool. Any questions on what's going on with mitosis here?
No,
Okay.
sounds good.
Okay, cool. So that is going to bring us to today's lab. So actually, before I get to that, I'm going to switch over to the lab assignment here. So I'm going to switch over our screen to the lab assignment. And so here, is everyone able to see the lab assignment now? Or maybe it'll take a little while.
Yeah.
Yeah, okay, cool. Okay, so the lab assignment is up. Oh, why is it paused? Resume share. Okay, so the lab assignment is up on Canvas. So if you go to the top of the homepage on Canvas, you'll see a lab PowerPoints and lab assignments underneath. Or I think it's on the second module. So you click lab assignment, lab 8 assignment, and you'll be able to download this Word document. And so this kind of shows you the instructions for what you're going to be doing for this lab. Okay, so the first thing you're going to be doing is activity 1. And so in activity 1, you're going to watch a video on mitosis. Okay, and so that's going to be this first link here. And as you watch that video, I want you to fill in this table, okay, in your lab books on page 62, okay, in your lab manuals. And you're going to fill in that table and draw in what is happening in each of these cells. Okay, so each circle represents a cell, and you're just going to draw in what's happening in each of those cells. Okay, and then on the right-hand side, you're going to write in a description of the major events happening in that particular event, okay, or phase. Okay. So you're going to do that for page 62. You're going to do that table, and so that's going to be located right over here. Okay. So again, you're going to draw in what you're able to see, and then write in the major events. Yeah, Paul.
Do we need to like scan that and send that to you?
Yeah, so you scan it if it's easier for you. So if you print out the whole document, fill it out, scan it. The other thing you can do is you can just kind of just type in what you would see. Either one works. I know it's a little complicated because I know not everyone's going to have scanners or an easy way to do this. So
there's an app.
Whatever works. Yeah. So if you have a scanner, you can go ahead and do that. If not, just tell me what you would see in that cell.
There's an app, I believe it's called ScanCam. You download it on your phone and allows you to scan everything.
Okay. Yeah, so I have heard of that too. So if there is that app that you can download, if you can type it into the chat, the name of that app, that would be great. So you can download that, a scanner onto your phone, and then you can use that to scan any documents and then upload it onto Canvas. And I'll show you how to do that as well. Okay. So you're gonna do that for all of the phases of mitosis except for prometaphase. Don't worry about prometaphase; it's not really that big of a deal. But you're gonna do that for mitosis. And then I also want you to watch a video on meiosis as well. This is what we're going to be going over in lecture next week, but there are a few questions at the end of the lab that have to do with meiosis as well. Okay. So watch that video, and then that'll help you answer the questions at the end of the lab. Okay. So that's activity 1. You're just going to be filling in that table. Activity 2, we're going to skip, so we can just ignore activity 2. And then in activity 3, I'll kind of go back to the, I'll show you what we're doing first. So in activity 3, you're going to be looking at various cells. And so you're going to be looking at some whitefish cells, and then you're going to be looking at some onion root cells that are undergoing mitosis. Okay? And so your job is to try to figure out how many cells you have in total, and then how many of those cells are in each of those phases. Okay? So I'm going to go ahead and go back to the PowerPoint so we can kind kind of do a few examples together. Okay. Okay. So that's that. Is everyone able to see the PowerPoint now?
Yeah.
Yeah. Okay. Okay. So what you're going to be doing, like I said, is you're going to count the number of cells that you have and try to identify how many are in interphase, how many are in prophase, how many are in metaphase, anaphase, and telophase. If you can get about 50 for each picture, that's great. If not, just use as many as you can. Then you're going to calculate the percentage of the cells that are in each phase. And again, I'll kind of go through exactly how to do that in a second here. Okay. So this would be an example of some cells that we're able to see undergoing mitosis. So these are some whitefish cells. And so here, we'll look at this particular cell right here. Okay. So in this cell, we're able to see our chromosomes, which are dark structures. Okay. These like dark black structures here. And our chromosomes are going to be lined up in a line. Okay, so based off of that, what phase of mitosis do we think we're looking at here? Anyone have an idea? So not anaphase. Yeah, so here we're looking at, just to kind of repeat that. So we have one cell right here. And so here we have our cell, this dark structure, this dark black components right here. Okay. These are the chromosomes. Okay. And so based off of the location of the chromosomes, which phase are we looking at? So Mary and Freddie are both saying metaphase, which is correct. So this is metaphase. So we see our chromosomes are all lined up along that metaphase plate. So we know this particular cell is in metaphase. Okay. Let's take a look at this cell over here. Okay. And so again, if we look at where our chromosomes are, we see some dark ones over here, chromosomes over here, and then other chromosomes over here. Okay. So that being said, which phase do we think this cell is in?
Anaphase?
Yeah, so this would be anaphase. Yeah, so this would be anaphase. And so we can tell because we have our chromosomes getting split apart. Perfect. So they're being moved to opposite ends of our cell there. Okay. This next one above, does anyone have an idea what we're looking at with this cell? What do we think? Yeah.
Is that telophase?
Yeah, so this is telophase. So telophase and anaphase can sometimes be a little bit hard to make out. But the way you can tell this is telophase is we're going to see these clumps of chromosomes all kind of gathered around each other. And then we're also going to be able to see that the cell has pinched quite a bit here. Okay? And so because it's pinched, we know we're looking at telophase as opposed to anaphase. Okay. Let's take a look at these cells over here. So again, this one, what phase would we be looking at over here with this cell?
Telophase? I'm not so sure.
So this one would be anaphase. Okay. And so this one looks a little bit different. Right, so Freddie's got some correct. So this is anaphase. So we can, again, see our chromosomes have been pulled apart. So we see dark structures over here and then dark structures over here. So they've been pulled apart over here. What about this phase, or this cell over here? What do we think this guy's in? Yeah, so Ryan's got this one right. So this is prophase. So this is prophase, again, just because we can see, we see our chromosomes all confined to a specific area, but they're not in a line, or they're not being pulled apart here. Okay, so this is prophase. Okay. I'm gonna show you another one. And so here we have this cell over here. Okay. In this cell, we can't see any chromosomes. And so what this tells us is that our chromosomes have not condensed yet. Okay, whenever we see the black parts, we know that our chromosomes have condensed, but this one's, we don't see any chromosomes because our chromosomes haven't condensed yet. So this tells us that we're looking at interface. Okay? So this cell would be an interface. This would be another cell in interface. This would be another cell in interface. This would be another one over here, another one over here. So all of these cells where you don't see any chromosomes in them, those are cells that are in interface. Okay. And then we'll kind of go through a few of them over here as well. So again, over here, we see some chromosomes being lined up in this cell. So here we're looking at metaphase. Trying to see a few others here. Again, we're kind of looking at cells in interphase. I'm trying to make out a couple other ones, but I think that's pretty good for what we've got here. Okay. So is there any questions on how to kind of determine what phase of mitosis we're looking at? Okay. All right, cool. Okay, so that being said, I'm gonna go back to this, our lab assignment here. And so here, what we're gonna be doing is you're going to be counting the number of cells that the number of whitefish cells that you're able to see in each of these phases, and the number of onion root cells that you're able to see in these phases. So we're going to be looking at both animal cells and plant cells in this lab. Okay. So so you're going to basically write into a table here. Okay. I only want you to fill out the number of cells individual, and then the number of cells, or the percentage of cells in each stage. Okay. So if you see, let's say maybe we count 65 total cells. Okay. You're going to write down the number of cells you're able to see. So let's say we saw 20 cells of our 65 in interphase. 15 cells out of the 65 were in prophase, 10 of the cells were in metaphase, 12 were in anaphase, and 8 were in telophase. Once you come up with all of those numbers, you're going to use those numbers to help you calculate calculate the percentage of cells in each of those phases. Okay? So 20 out of 65 cells gave us about 31% of cells in prophase. 15 out of 65 gave us about 23% of cells 31% of cells in interphase, 23% of cells in prophase, 15% of cells in metaphase, and so on and so forth. So you should come up with about 100%. Okay. So up here, I have an example. And so here, again, we're looking at some whitefish cells. And so again, we'll be looking at prophase. These ones over here show anaphase or metaphase. This one shows anaphase, and then we see telophase over here. Okay. So this is just an example to help you kind of orient yourself as to what phase of the cell you're looking at. One thing I do want to bring up is that you don't need to come up with exact numbers. So if your numbers are off a little bit, don't worry about that. This is more, I'm not going to be grading you on whether you get the right numbers. The only thing I want you to be doing is just getting comfortable and practicing looking for cells in specific stages. Okay. Again, you don't need to worry about getting the numbers exactly right, because I don't really care about that. It's more just for you to be able to see kind of the trends in each of those phases. Okay. I do want you to skip this row here that says number of cells group. Okay, just skip that one. Don't worry about it. It's not going to mean anything. Okay. So you're going to do the number of cells per individual, or of the individual, and the number percentage of cells in the total group for both the whitefish cells and the onion root cells. Okay. So these are the cells you're going to be using. So this is the whitefish cells. Okay. Again, if you can count 50, that's perfectly fine. If you want to count the whole thing, you can do that as well. It's totally up to you. And so again, try to identify how many of these cells you're able to see in each of those stages. Okay. And then here's the cells you're going to be using for the onion root. Okay. So you're going to be using this to help you try to, again, identify the different phases of mitosis in our plant cells here. Okay. And the last thing you're going to do is you're going to answer the extra questions on page 64. Okay. So what you're going to be turning in or uploading. So Ryan says, so these slides we need are in the lab document. So the slides that, so these slides, yeah, so the slides that you're going to need are in the lab document, right? So these are going to be the slides that you're going to be using. Okay, these ones here. Yeah. Okay. Okay. So you're going to be turning in this page from here on, which is page 4, all the way down to page 6. Okay, which is over here. Okay, so on page 4, again, you're going to be writing in the major events that are going to be happening during each of these stages of mitosis, and then draw in what you should be seeing in your in all of these cells. If you're not able to draw it in, then you can just write what you should be seeing, and that's totally fine too. Again, here are the whitefish cells that you're going to be using. Here's the onion root cells, and then you're going to list them into this table here. You're probably going to have to, if you're going to type it in, you'll need to make a text box and then just add the text box over the picture over here, over the diagram, and that'll be fine. And then there's some room over here for you to answer the extra questions on page 64. Okay. So there's a total of 6 questions there. Some of these questions have to deal with mitosis, and some of them have to deal with meiosis. So you will need to watch that video on meiosis in order to answer some of those questions. Again, if you don't get the right answers to those, don't stress it too much. Don't stress too much because we haven't gone over meiosis. We'll go over that next week in lecture. But those are going to be the areas where you can write in your answer for the extra questions on page 64. Okay. So once you are done with this, you're going to save it as either you're going to save it and then scan it. And then what you're going to end up doing to submit it is you're going to go to, let me go to our Canvas page here. Okay. So you're going to go to Canvas. You're going to go over, you're going to go over to the assignment section. And when you go to the assignment section, you'll see Lab 8, Mitosis and Meiosis. You're going to click that. And then, once this comes up here, any second now. Any minute now. Sorry, my internet's really slow today. Okay, so on the top right-hand corner, you will see a button eventually that says Submit Assignment. Okay. So you're going to click Submit Assignment when you're completed with the lab. You're going to hit Submit Assignment, and then it'll ask you to choose the file to upload, and then you'll just upload the file through Canvas that way. Okay. If you are unable to do it through Canvas, you can always email it to me, either through Canvas, through the inbox, or you can email it directly to my Gavilan email, and that's listed in the syllabus as well. Yeah, Paul.
I just wanted to share, I recently found out if you have an iPhone, when you go into Notes and you go to the camera mode, there is a scan function, and from what I understand, it converts it to a PDF, and then you could just put, put it in your drive.
Oh, nice.
Yeah, I found it. I know it's there, but I haven't scanned anything yet. But that, in theory, it should work. Okay.
Yeah. So that's another way you can go about it, and then you can always upload it from your phone through Canvas as well. So that will work too. There's a few ways of going about submitting it. If you have any other questions on how to submit it, feel free to email me or send me a message, and I can clarify that a little bit for you.